Good afternoon. Welcome to the AIRA Matrix Sponsor Symposium. My name is Pedro Oliveira, and I'm a consultant in histopathology at the Christie Hospital in Manchester, UK, one of the largest cancer centers in Europe. I will share this presentation with Nitin Singhal, Head of Products from AIRA Matrix Mumbai, India, and our subject today is the use of artificial intelligence in the radical prostatectomy specimen. There is no doubt at all that the future of pathology is going to be digital. And artificial intelligence offers a tremendous opportunity to improve routine histopathology diagnosis. But for that, it needs to fulfill what I call the toaster criteria. It needs to be something that survives testing. It needs to be something obvious and awesome and particularly simple. We don't like complicated things in pathology. We use a simple methodology, the microscope. It needs to be trendy, essential, and required, something that we need to use like a toaster on an everyday basis. So why I think that radical prostatectomy fulfills and is an optimal example of a specimen where these features need to be applied. As you know, as pathologists, what we do is issue a pathology report. We write something. And this document is transmitted to the clinician that is going to be using it for basing the clinical decisions. What we do to obtain a pathology report is to transform the specimen in a number of glass slides. As you see here, at Christie, we use wall-mount slides for radical prostatectomies, and depending on the size of the prostate, we have normally a medium of 10 to 12 of these large glass slides. When we are issuing pathology report, it is expected that pathologists will going to comment some findings, namely the Gleason patterns, primary, secondary, tertiary, the percentage of each Gleason pattern, then the grade group. In addition, it is expected also that we comment the volume of tumor, the location of it, and also more recently, the amount of intraductal carcinoma and cribriform morphologies. So here we have the outcome of our radical prostatectomy specimen, and I just label at red the slides that show presence of tumor, and this is what we see at the microscope. But as we all know, prostate adenocarcinoma, it's quite diverse, and from one slide to another, we can have different patterns. And this is an example where in two slides, we have two different patterns, Gleason 3 and Gleason 4. So the question is, what is the methodology that we pathologists use to achieve the results that we need to express in our document for the clinician? As you are aware, we use eyeball assessment. We just look through the microscope to the slides. But attention, our eyes are not independent. They are just an extension of the brain. And as such, we suffer from cognitive bias, subjective assessment, knowledge limitations, inaccuracy, and also it's difficult for us to deal with very complex cases. So of course we can have methods to mitigate this visual estimation. And for example, one of it is to use charts to help on estimation of the proportion of mixtures in a similar way that the geologist does for assessing the mixture and the content of different minerals in rocks. But let's be realistic. If we see this slide, who is going to be comfortable in estimating the percentage of black and red dots? At best, we can say, we guess, we do our best, and we get a result. But that result is inaccurate. But even if it is inaccurate, it's interesting that all our knowledge, that means we use it for prognosis, therapeutic decisions, clinical trials, and also multiparametric MRI correlations, in spite of knowing that this is at best a guess. So what we are having is a sort of big data, but with somewhat some flawed data. So how can we improve? Of course, there is only one solution, is to use artificial intelligence algorithms. The benefit of the artificial intelligence and software is that there is no cognitive bias. They are very objective in their assessment. They have unlimited knowledge capacity. They are precise on the assessments, and they can solve easily any complexity. And so we reach big data. And we need a software that calculates automatically the Gleason patterns, percentage of each Gleason pattern, grade group, volume of tumor, location of tumor, even the presence of IDC and cribiform morphologies, and present it us with the hard work done. Then, for us, this ancillary method is going to be either validated or we will change partially, and with time, we will know that the software will learn how to solve minor problems. This area for radical prostatectomy has an enormous potential because people think, or let's say, what is at stake is that does not refer only to pathology. For example, with this data, you can enhance multiparametric MRI, and you can use and improve it on screening, on diagnosis, on therapeutic decisions, with consequent lives and costs savings. The same can apply to robotic surgery. Our data could populate 3D imaging location, allow robots to faster performance, to be more accurate, so we also can have better results with this data. And last but not the least, pharmaceutical industry. The data that we will be gathering with artificial intelligence on the analysis of radical prostatectomy specimens can be used to drug development, to create biomarkers for easy identification of prognosis, to populate clinical trials, and to allow us precise, tailor-made patient selection with consequent cost savings. So with this in mind, we start a collaborative project with AIRA Matrix to try to develop an algorithm for analysis of the radical prostatectomy specimens. And at this point, I will give the stage to Nitin, who will go through the technical aspects of this software.
Thank you, Dr. Oliveira. Good afternoon, everyone. I am Nitin Singhal, and I look after products at AIRA Matrix. Let me begin with a brief introduction to AIRA Matrix. We are a technology company that provides deep learning-based image analysis products and services for the healthcare and pharmaceutical industry. AIRA Matrix was founded in 2011 and has been working in histopathology image analysis for close to a decade now. We are a team of 60 with expertise in different areas like toxicologic pathology, oncopathology, machine learning, deep learning, data analytics, high performance computing, and productization. We collaborate with pharma companies, CROs, and oncology research institutes worldwide on an array of image analysis solutions. We develop AI-based image analysis and image management products and solutions for toxicologic pathology, discovery pathology, oncopathology, microbiology, and ophthalmology. We develop a range of applications from screening and triaging to disease quantification and to prediction. As a research-centric company, we are constantly pushing the boundaries of the state of art in computer vision. At AIRA Matrix, AI research is practiced as a multidisciplinary exercise where insights from data-driven machine learning, deep learning are fused with domain-specific knowledge to amplify the quality and causal accuracy of the predictions derived. We are creating state-of-the-art perception and reasoning capabilities for our AI technology to observe and understand the contextual meaning. Our capabilities in computer vision, machine learning, deep learning, knowledge representation, and reasoning are used to predict required information robustly. Some key challenges we tackle include lack of sufficient labels needed for traditional supervised learning approaches, the need to ingest and link multiple data modalities, efficient deployment of complex deep learning models, and the need to build AI solutions that are interpretable. We are developing few shot, zero shot, class imbalance learning systems that learn from minimum labels to predict behavior. We are creating virtual staining system that can produce virtual immunostains from existing H&E whole slide images. To overcome deployment challenges, we develop knowledge distillation techniques that effectively learn a small student model from a large teacher model. Our researchers and engineers are constantly aiming to apply the state-of-the-art solution in different healthcare aspects, for example, improve disease diagnosis, augmenting pathologists by increasing workflow efficiency to save precious time. Now to the prostate carcinoma. Our journey on prostate carcinoma started with top finishes in global Gleason grading challenges. We began by designing a deep learning framework for Gleason grading in tissue microarray, followed by refining and validating the framework on needle core biopsies. We achieved a significantly high agreement with pathologists grading and scoring in both of these challenges. In the current project with NHS Christie, we are translating our AI framework to comprehensive histopathological reporting of radical prostatectomy specimens. One of the main challenges we encountered while transferring or retraining our deep learning models was to generate a labeled dataset. A single RALP image has close to 30,000 to 50,000 relevant glands, which are incredibly cumbersome to mark manually. To circumvent this challenge, we have developed a continuous learning framework. The pre-trained model from needle core biopsies is used to generate rough annotations on RALP images, which are later refined by the pathologist, and then the training continues. This process repeats 3 to 4 times till the model converges to the desired accuracy. The figure shows the main step that constitutes the algorithm pipeline. Some key elements of the algorithm include a multi-resolution ensemble of segmentation models to delineate tumor region and identify Gleason grades 3, 4, and 5. Stain normalization to reduce the color and intensity variation present in stained images from different laboratories. We have designed a novel approach for the stain normalization problem using fast neural style transfer coupled with adversarial loss. The approach was presented in just concluded MICAI 2020. Stain normalization typically increases the prediction accuracy of our AI system. To improve upon the segmentation performance over the state-of-the-art fully convolution network, we present here HistNet or histopathological network, which uses an encoder-decoder architecture similar to UNet as seen. The encoder network incorporates inception-like modules which provide the capability to aggregate context to extract complex features for identifying tissue cell morphology as the feature maps are downsampled. HistNet allows better propagation of information from lower resolution layers of the decoder to higher resolution layers using a dense shortcut connection at each decoder stage. This concatenates information from the previous stage with lower resolution with the output of the current stage convolutions. Additionally, both encoder and decoder block uses proposed parallel convolution to enhance content and context aggregation further. The proposed architecture achieves better segmentation performance than most state-of-the-art semantic segmentation models while using a significantly lower number of parameters. Similar to the inception block, which combines information from different scales, HistNet encoder uses a modified inception block, what we called as inception plus block, as seen on the left. Inception plus block consists of 4 branches to gather information from different scales of the receptive field. The output of the branches is then concatenated with input feature maps. Importantly, 2 branches share a convolution operation as this allows more efficient parameter use. A convolution 1x1 layer is used to control the number of feature map channels after the concatenation. Inception plus block uses dilated convolution in parallel to standard 3x3 convolutions. The dilation rate of the convolution adds further ability to extract multi-scale information. To improve the model's ability to finely delineate morphological features on whole slide images or whole mount images, we improve the decoder to have higher context information. For this, we use a dense shortcut connection at each decoder stage, which concatenates information from the previous stage with lower resolutions with the output of the current stage convolutions. This is beneficial as it allows the model to reuse information extracted from lower resolution stages, providing efficient pathways to information propagation, effectively providing multi-level feature fusion. Furthermore, similar to encoder blocks, these modules also use dilated convolution in parallel to standard convolution. Let's look at the results of our tumor identification model. The figure shows an example comparing the manually delineated tumor region on the left with the AI predicted region on the right. The results are shown at 2.5x magnification. The results show a very high level of concordance with the manual markings. Here is another example of tumor identification using our algorithm, illustrated at a higher magnification of 10x. As seen, the algorithm is capable of identifying all the glands with very high accuracy. Here we show the qualitative performance of our algorithm and the percentage area of each of the Gleason grades. Besides the core level score and quantification, the system also outputs pixel level overlays. Pathologists can use these overlays as a second read to confirm that no area from the image is missed from analysis, in addition to improving the accuracy and precision of Gleason grades. The illustration here shows another example of Gleason's grading output at a higher magnification of 10x. As can be seen, approximately 66% of the tumor region was identified as Gleason grade 3 and the rest 34% was identified as Gleason grade 4. Our next step in AI-based RALP reporting is to develop 3-dimensional reconstruction of the radical prostatectomy specimen followed by reporting of volume estimation of the specimen, prostate, and tumor region. As an extension, we will also be looking at identification and density measure of perineural invasion and assessment of extraprostatic extension and seminal vesicle invasion. You just witnessed our journey that had started with histopathology reporting in the previous slides. Our next goal is to translate AI insights from histopathology to multimodality data from MP-MRI, biochemistry, proteomics, and genomics. Eventually, this will lead not only to improved reporting but achieving better treatment outcomes. As AIRA Matrix envisions, artificial intelligence has opened up infinite possibilities for life sciences. On that note, I would like to end my presentation and declare the forum open for any questions.